아,
Guys, sit down, sit down.
Everyone sit down.
We're gonna be starting now. We know some other people are supposed to come, but we'll start now due to time constraints. Thank you all for coming to the GIDAS Symposium. GIDAS, which stands for Genes and Diseases and Symptoms, is a Wednesday lunch club that focuses on genetics and biology. Over the year, our freshmen did research on the GEO website, and they'll be presenting it today. So first, we have Amy Kim with her topics on hemophilia A. Round of applause.
Hello, I'm Amy Kim in grade 9, and today I'll be talking about a genetic disorder called hemophilia A. So, hemophilia A is a genetic bleeding disorder caused by a deficiency or low levels of blood clotting protein called factor VIII. Factor VIII is the key protein needed for blood clotting, and liver sinusoidal endothelial cells are special cells in the liver's blood vessels that produce factor VIII needed for blood clotting. So if I describe the difference between someone who has hemophilia and a healthy body, it's that a person with hemophilia will have lack of clotting factor, which means they'll have a continuous loss of blood. So my article talked about a few ways to treat hemophilia. The first one was the intravenous injection of the F8 protein, which is a traditional method still used today. Next is gene therapy, which proved to be not very effective, and also cellular therapy, which showed that there is an effectiveness in treating hemophilic conditions in mice, which shows potential for possibly treating that for humans. My hypothesis was that since fetal LACCs shows more effectiveness in factor VIII production in mice through cellular therapy, then there is a difference in gene expression for fetal and adult LSECs that may be applied for factor VIII production in humans. My data shows that there is an upregulation and downregulation between adult LSECs and fetal LSECs. This graph shows that there is an upregulation and downregulation between fetal LSECs and other ECs. So graph 1 shows that adult and fetal LSECs have different gene expressions, and graph 2 shows that fetal LSECs and other ECs also have different gene expressions. So my conclusion is that cellular therapy is a potential method in the future to treat hemophilia A, and there will be a difference in the effectiveness of fetal and adult liver sinusoidal endothelial cells and other endothelial cells in cellular therapy due to different gene expressions. Thank you.
And thank you. And next we'll have CJ Park with his topics on cystic fibrosis.
Hello, my GEO is about cystic fibrosis. Underexpressed and overexpressed genes in cystic fibrosis study. So first off, in this study, there's 6 macrophages were collected from 6 CF patients and 6 non-CF patients. Macrophage is an immune cell that is in and this explores its role in chronic inflammation. This study focuses on transcriptional diversity and macrophage repolarization and how chronic pulmonary disorders like CF have different implications. The macrophages were first polarized to an M1 state, which is an anti-inflammatory state, using LPS. Then it was repolarized to an M2 state, which is a pro-inflammatory state, using these 5 different methods. First off, I'd like to talk to you about what underexpressed and overexpressed genes are. Underexpressed genes are genes that have low activity or low mRNA levels, and overexpressed have high mRNA levels and high gene activity. The underexpressed genes in this study showed a significant downregulation in the M2 state. There were reduced pro-inflammatory responses and enhanced toleration to repeat LPS challenges. And in overexpressed genes, there were upregulated in M2 state. So it has enhanced phagocytosis and efferocytosis. Phagocytosis associated pathways in IL-10 stimulated macrophage, and potential therapeutic targets for chronic inflammation and CF may occur. As a conclusion, macrophage repolarization includes significant transcriptional changes and further exploration of gene targets for therapeutic inventions, and maybe some clinical setting validation is needed. Thank you.
Next, we have James Kim and his topics on progeria.
So I made my research on progeria smooth muscle cells. So progeria occurs in 4 to 8 million child, and it's about premature aging. So how this works is that in normal cells there is a protection layer called SMC, and this prevents the degradation of arteries, while in progeria cells SMC is evaporated, so there is a degradation of the arteries. So my hypothesis is that in progeria smooth muscle cells, specific genes exhibit significant differential expression when compared across the groups A, B, and C. So this is my data. So as you can see, this is one of the genes in here. So you can see that the gene is overexpressed in group C, this one in A, and this one in B. So these are one of the 4 genes that are underexpressed and overexpressed. So these genes were largely involved in overexpression and underexpression process that's showed in the volcano plot. So my conclusion is that my analysis identified key overexpression and underexpression genes in progeria smooth muscle cells, and this highlighted their potential role in disease progression. These findings provide insights into molecular mechanisms, and they serve as targets for future therapeutic research.
Next, we have Mary Cho and her topics Fragile X syndrome.
Hello, my name is Mary Cho, and I'll be introducing about Fragile X syndrome. So first of all, Fragile X is one of the most common causes of inherited intellectual disability, and is an X-linked genetic disorder. Some symptoms are distinct facial features, delayed speech and language development, autism spectrum disorders, learning disabilities, and cognitive impairment. So, Fragile X syndrome is caused by a mutation in one of the gene called FMR1, and see there is a expanded region in the gene where there's CGG repeating, and methyl group is attached to the CGG repeats, and as a result, FMRP is not produced. My hypothesis is that genes are underexpressed to induce Fragile X syndrome. So these are the graphs that I got, and the first one is called UMAP plot, and as you can see, it is separated into two groups, which shows different genetic expression profiles, and the second one is called box plot, and the boxes are generally at the bottom, which means that the genetic expression are primarily underexpressed. These are the significant gene names. And my conclusion is that although the genes of Fragile X syndrome are underexpressed, primarily underexpressed, genes are either underexpressed and overexpressed because the expression of the FMR1 gene depends on the CGG repeat size. Because of underexpressed in full mutations and overexpressed in pre-mutation carriers. Thank you.
Next, we have Roy Kim and his topics Alzheimer's disease.
Using antisense oligonucleotides to reduce Alzheimer's disease risk. So, as a lot of you may know, Alzheimer's disease is a neurodegenerative disorder that causes memory loss, cognitive decline, disorientation, personality and behavioral changes, and language problems. The causes of Alzheimer's are not fully known, however, major factors include genetic risks like APOE genes. Apolipoprotein E, specifically APOE4, plays a crucial role in the development of the disease by influencing the accumulation of amyloid plaques in the human microglia. Amyloids are proteins that form insoluble structures that usually lead to disease. Amyloid plaque buildup in the human microglia can cause miscommunication between neurons, inflammation, the disability to clean waste, and more. This and other factors are major contributors to Alzheimer's disease. Antisense oligonucleotides, or ASOs, are short synthetic strands of nucleotides that are complementary to specific regions of messenger RNA. These molecules can bind to these mRNA and reduce amounts of protein production. In this study, scientists found ASOs that reduced levels of APOE4 amyloid production. They hypothesized that if these ASOs could reduce amounts of APOE in human microglia,
then it could reduce risk of Alzheimer's disease. Scientists studied these ASOs in both lab-grown human microglia and in living animal models with human microglia. In the graph, the red dots show treated human microglia and shows that it is overexpressed, meaning it produced a lot of protein, while the treated blue dots show that the protein was reduced. Scientists concluded that ASOs can affect APOE4 and their production of amyloid, and overall the risk of Alzheimer's disease. Although the study does not directly stop or prevent Alzheimer's, it could have implications for Alzheimer's disease prevention and treatment. Thank you.
And lastly, we have Thomas Kim with a presentation on Down syndrome.
Hi, my name is Thomas Kim. Okay, so my research paper was about utilizing apigenin to treat Down syndrome. So, what is Down syndrome? So this is a karyotype of a patient with Down syndrome, and as you can see here, there's an extra pair of the 21st chromosome, and this ultimately causes intellectual disabilities and motor disorders. And the research team investigated this substance apigenin as a possible treatment of Down syndrome. So this is a molecular structure of apigenin, and it is a plant compound found in various herbs and fruits, and it is known to ameliorate the effects of Down syndrome from protecting neurons from oxidative stress to anti-inflammatory effects. So this diagram shows one possible effect of apigenin in the body. So as you can see here, in normal human lymphocytes, it has no DNA damage, while in breast cancer cells, it takes various measures to ultimately cause apoptosis in those cells and ultimately have a chemopreventive effect on the body. So anyways, these are my GO2R results. Okay, so as you can see here, these are all like the volcano plots of expressive genes, and as you can see, like these common outliers in the right or left, these are all the overexpressed or underexpressed genes. And I just selected these 5, and these genes were largely involved in the overexpression and underexpression process. Okay. And also, this is a bit different, but this is the top differentially expressed genes. And as you can see, like the first one is GC21, so it's from the 21st chromosome. So as you can see in the graph 2, it has various varying effects on different groups as well. And what does this implicate? So ultimately, apigenin had it inhibits pro-inflammatory genes such as the NFKB protein, and it also has some anti-inflammatory cytokine effects, which ultimately could reduce the effects of Down syndrome in the patients. Yeah, so that's it.
Thank you for attending the GIDAS Symposium, and that will be it for today. Thank you.
Don't we get to ask questions? I have a question. amazing. Like, all of those words that you are all saying are extremely long and difficult to pronounce, and well done on doing that. And obviously your research is really well done as well. I have a question for Roy. Sorry, Roy. But when I've looked at like Alzheimer's disease, some people call it type 3 diabetes. Have you seen anything like that?
No.
In your research? Okay. That was my question. But anyways, congratulations everyone. It was really well done.